Hola amigos, my name is Dr. AVISHAY SINTOV and I'm very happy to talk to you today. I'm very excited to talk with the people of Argentina. As a son of an Argentinian, Argentina has a very warm place in my heart. So let me first introduce myself. I got all my degrees in mechanical engineering from Ben-Gurion University in the Negev in Israel. After that, I had two postdocs, one in the University of Illinois and one in Rutgers University. And since 2020, I founded the robotics lab at the School of Mechanical Engineering at Tel Aviv University, and I lead a group of about 2 PhD students, 11 master students, and some undergrad students. This is only part of the team. And we work on a lot of exciting projects. And I would like to, first, I would like to talk about 2 interesting projects that we work on. But first, let me talk about research in robotics. So research in robotics is a multidisciplinary area where we deal with a lot of aspects of the robot. From the design, meaning how will the robot look, what is its structure, material and stuff. After we have that, we talk about how can we model this robot in terms of how it works. And with the model, we can do motion planning. We can do some interesting artificial intelligence and machine learning algorithm, and we can control it in order to move. So this is a lot of aspects of the robotics, and we in the robotics lab in Tel Aviv University work from the entire spectrum, from the design, how it looks, to plan the motion and do some interesting algorithms for artificial intelligence. So I'd like to talk to you about 2 aspects and 2 projects in my lab. One is human-robot interactions. How do we have robots collaborate with the humans in shared tasks? And later I'll talk about printable hands. So human-robot interaction, this is the future. I mean, we want robots to help us do hard stuff. We want them to help me and do the dishes, and we want them to clean the toilet, but we want them also to assist me in everyday tasks or help the disabled or help a doctor in surgeries. And we need to do this in an intuitive and natural way. Let me explain. So when 2 humans work together, they can do shared tasks even without verbal communication. I can look at the motion of my counterpart human. I can observe what he's holding. I can predict his trajectory. And with my perception, I can act to assist him. For example, open a bottle. If he's had difficulties, I see him holding a bottle, I can assist in opening the cup. So we want a robot to do this. We want a robot to understand our natural and intuitive motion as we do every day and assist us in these tasks without interfering. We don't want the robot to interfere. We want them to help. So this is the idea. We want a robot to assist us in a way humans do. So some solutions to try to understand what humans want can be electrical myography, where we sense muscle signals from the arm or from the leg or something like that, called EEG. But that requires some extensive hardware. It's a lot of noise, and it's not that easy to extract. Computer vision also, we put cameras around us and try to estimate the motion of the human, but some occlusions may occur. Lightning, we have many problems with that. And some even try to apply gesture recognition. When we do some kind of gestures, it's not natural. We want a robot to do something, we give them a predefined gesture, and the human needs to train in order to be able to control this. Other applications, such as sensing brain activities, we put some kind of helmet and we try to measure brain activities and try to analyze those patterns. But these all solutions are unnatural and very hard to adapt to. Force myography, on the other hand, is easier application. Force myography is the measure of muscle perturbations on the let's say the forearm. We measure the perturbations of the muscle while we hold stuff, while we do tasks. And previous work, not my work, have shown very good results in understanding these patterns. When we do, mostly in tasks for rehabilitation and trying to assist disabled in doing more stuff. But we want to take this and to apply it to human-robot interaction, use force myography to measure, to try to analyze motion. So we built this prototype, these prototypes here. These have 15 force sensors, very cheap sensors, less than $1. And we put it on the arm and we collect data while doing stuff. So the first goal was to try to predict or understand what a human is holding. For example, whether I'm holding a scissor, so the task will be to assist in giving a paper, something like that. If I'm holding a plate, so I want to eat probably. This gives us a lot of information on what the human wants. So this was the first task, and understand what do we need in order to predict these objects by a single user. And we got very good results. We have some iterative algorithm where we can predict over time. We try to analyze where is the most important places to measure. We got a 97% success rate in predicting. So I mean, if I'm holding a bottle, there'll be 97% chance that I'll predict it correctly. The problem here with this work is that we did it for one user. When we applied to a new user, we got very poor results. So we tried, the next step would be to try to train on multiple users and try to train a neural network model based on a lot of data from many objects and many users. So we took a lot of objects, very different objects. We have a series of bottles and a series of plates and a series of drinking cups. And we had the humans do a lot of manipulations on them. And we recorded the force myography and signals while doing so. So now we try to analyze these measures based on a lot of grasp of the object, not just task-based grasp. And we did it for many, see, here you can see different users. And we took all this data and trained a very sophisticated model. And it got very good results, about 98% success rate. And you can see this test in real time. You can see it's a new user. Just we found someone around the corner and we asked him to assist. And you put it on, and you can see very good results in predicting the object that he was holding while manipulating the object in real time. This is kind of very, very interesting. Okay, sorry. So what can we do with this? Again, human-robot interaction. We have information about the object, so this gives us a lot of information about what the human wants. And then we can do pose estimation, understand how the human is holding the object. That is the next step. Also, we want to project the trajectory of the human. How is he moving in real time? So in earlier steps, the robot can already act to assist, so the human won't have to wait for him. So we want natural motions. Another option is telemanipulation. So unfortunately, we live in times when we need to treat a patient in quarantine remotely. So we want robots that can work there, but operate. A doctor or a nurse can operate from far along, but they don't have time to train for different joysticks and touchpads and stuff like that. We want them to do natural motion. So we put these wearable devices on their hands, and they do natural motions. It's amazing. It will be a very good progress. and they can use virtual reality headset, and it's almost like they are there. Okay, next step I want to talk about printable robots or printable robotic hands. So in factories today, many tasks are done manually. And why is that? So for example, in automotive industries, dashboard and stuff like that require precise manipulations, require human operation. Also packaging in Amazon, many operations still require humans. And we don't want to replace humans, but necessarily we want to assist them. We want affordable hardware. And the reason we don't have many robots doing this stuff is because robotic hands can do precise manipulations are very expensive. These are commercial hands. The price moved between $50,000, the cheapest, to about $50,000. That's crazy. You cannot fill your factory with these hands, you'll get bankrupt. So another reason is that if you want to do very simple tasks with these hands, for example, this seems simple, but it requires a lot of engineering in terms of making sure that the grass is stable and you don't drop the object, and you don't break it. So it requires a lot of, it's not easy. It seems easy, but it's not easy. On the other hand, printable robots, underactor, I'll not talk about the underactor robot, but robots that are printable and very simple to something like, so printable robots are this is not new, but we want to apply it to robotic hands. Okay, so it's not my work. Yale has a very nice project about printing hands. I mean, print hands. You can go ahead, just link, download the hand, print it, and you have a very nice hand. The problem is, and they work great. They can grasp objects and adapt to their shape, and they work amazingly. The problem, if you want to do fine manipulation or something like this, it's very hard because these hands are printed and there are a lot of errors and they're not precise. So we don't have a model for these. And we want to do a lot of manipulation, but we don't have a model for this. The problem is we need to model. And so, and they're also non-linear, but the details are important. So what we do is we do database, we do machine learning. We collect a lot of data on these manipulations, and then we take this data and build models. And we see that we can do very precise manipulations based on these models. And we can take these hands that are very good for grasping and also do precise manipulations while their hardware is very, very cheap. And then we can do a lot of, test these manipulations. And of course, we want to do this in 3D. And another challenge is how can we take these hands and print them as one unit? So if until now they are made a lot of parts, and someone that doesn't have a lot of technical work, technical experience will have a hard time assembling this. We want them to be printable. As one unit, we print them and they work astonishingly. And then we can miniaturize them and do them for medical procedures or something like that. I'd like to thank you very much for having me, even if it's virtual and recorded, and hopefully in the future we'll be able to meet in person. I'd like to give credit to my students who are working on these projects. Please feel free, if you have questions, please send me an email, or you can go into my website. Again, thank you very much.
Hello, everybody. Buenos dias. My name is Gazit. I'm a faculty member at Tel Aviv University, involved with the Blavatsky Center for Drug Discovery, Faculty of Life Sciences, as well as the Faculty of Engineering. Today, I would like to speak with you about something that I see as the forefront of science and medicine in years to come. And let me just share my screen. Here it is. And the topic is metabolite medicine. Don't worry about it. If you never heard about metabolite medicine, I promise you that by the end of my presentation, you'll be all aware of it. We study aging, and aging is a normal process, but we would like to think about those situations in which aging could be a healthy one. As some of you may know, I've been involved in more than 2 decades in the study. I also recently received an award on this. And when I'm not doing science, one of the things that I like to do is to do sports. And I'm proud of my ability to do some pull-ups also at the age of 55. But I really like to see this movie that you see also on YouTube, in which you can see this guy. He's 90 years old, 90, and he can do few tens of pull-ups with not much problem. So how could it be? Why one person could not live up to the age of 90 and another person could do pull-ups at this age and be quite healthy? So you may say that it's all genetics. There's nothing to do about it. Everything is written in our genes. So quite a lot is related to genetics, but it's not everything. Another example that I like, and I guess that many of you are familiar with, is the story of the twins. This was quite a famous movie about 3 identical strangers. These were twins which were brought up in a different environment. And you could see that when they were young, they looked quite similar. But while aging, one of them did not survive, unfortunately. But the others, you can see they don't look just the same. Personally, I'm interested in the reason that one person has more hair than the other, and advancing age pull-ups does not help you to keep your hair on. But clearly there's something that is making the difference. And it's not only the genes, not only the genetics. One of the hints towards the different processes and aging versus healthy one versus non-healthy one relates to some diseases that we consider to be more prevalent in old age, like arthritis or cancer, lung disease, renal diseases, and neurodegenerative diseases. But I think the 2 diseases that are mostly associated with advanced age are cardiovascular disease and Alzheimer's disease. This is very uncommon, unlike, unfortunately, cancer that sometimes you see with children. It is very uncommon to see those diseases in children. But this is not always the case. Already in the 60s, Dr. McCauley had identified children who had strokes, heart attacks at the age of 8. And what was with these children? They had abnormal concentration of a metabolite that is known as homocysteine. He published a book that got quite a lot of attention called the Homocysteine Revolution. Other people who were following up on his work called homocysteine as a new cholesterol. It allowed to explain all kinds of different diseases that are related to advanced age. Even much more recently, in 2018, there was a consensus statement that relates homocysteine levels and dementia. I will read. We have presented a consensus statement and concluded with elevated plasma, a total homocysteine is a modifiable risk factor for the development of cognitive decline, dementia, Alzheimer's disease in older persons. And it relates to some variation in the gene. So what is homocysteine? Homocysteine is a metabolite. It's one of those end products of metabolism. You can find thousands of them in the human body. They are small, involving various activities, structure signaling, stimulatory, inhibitory effects. And there's something that we call the metabolome. The metabolome is the complete set of thousands of metabolites that we have in every biological cell, tissue, organ, or a full organism. When metabolites were found to be related to some disease state, it connected to other very central research that is done in our lab, which is related to molecular self-assembly. We studied the organization of small molecules like these metabolites into ordered structures. And in an illustration, it could look like what you see here, the organization of the magnetic disk. So we try to understand how molecules, like the metabolites, organize to form ordered structures, like you see here, by the process of what we call self-assembly, the organization, the spontaneous organization of molecules to form ordered structures, like you see here. For some non-expert, I call it sociology of molecules. We try to understand, just as how people organize into ordered structures, how molecules can arrange themselves into ordered assemblies. And the importance of this organization is the fact that many times the organization of molecules into these ordered structures is related to disease. Already a few years ago, we established the ability of metabolites to form ordered assemblies by this process of self-assembly, which I just described. And it's a new paradigm for inborn error of metabolism disorder. Disorders in which children have very high concentration of metabolites, like those children who have high concentration of homocysteine and got heart attacks and strokes at the age of 8, and other diseases like phenylketonuria, PKU, in which children have high concentration of phenylalanine. This is the disease which is diagnosed for every newborn when you pinch the toe and determine if he or she do have PKU. And we demonstrated the ability of the formation by self-assembly of the metabolite structures in the brain of these patients with PKU, phenylketonuria. The understanding of the role of metabolites in aging and in age-related diseases is becoming more and more prevalent. This is again a quite recent paper from 2019, try to understand various metabolites and their role in Parkinson's disease, another disease that is related to old age, and it's quite complicated type of activity, but altogether, what we understand that there's some change in metabolites, even with people with identical genetics. You can have different set of metabolites, and which could be related to various diseases, Alzheimer's disease, Parkinson's disease, cardiovascular diseases, and others. Based on the understanding that metabolites could form ordered structure by this process of self-assembly, and the understanding that there's correlation between abnormal concentration of metabolites and those diseases of old age, especially the degenerative diseases, we suggested already not long time ago the possibility that proteins that are involved in neurodegenerative disorders like Alzheimer's disease, Parkinson's disease, and others may be seeded. The metabolite structures can affect the protein to form all kinds of deposits. And this can explain some clarify some unexplained epidemiological associations that were established in the past. And indeed, a few years ago, we understood the role of the formation of structures of quinylonic acid, which is a metabolite that is accumulated in Parkinson's disease, and the formation of plaques in the brain of these patients. And very recently, we went back to this homocysteine, which I started all the discussion with those children who unfortunately have the high concentration of this metabolite, and establish its crosstalk with Alzheimer's disease beta-amyloid polypeptide, a polypeptide that form plaques in the brains of Alzheimer's disease patients. And this was just published recently. And I don't want to go into too much science, but this is slices from the brains of model mice to understand Alzheimer's disease. And you can see the colocalization in the brain of the metabolite assemblies together with the amyloids, the structure that have been accumulated in the brains of the patients. The aggregation of metabolites and the formation of all the structures occurs also in other diseases. Many of them are more common in advanced age. Kidney stones, gallstones, crystals in gout, plaques, cardiovascular plaques, and amyloid brain deposit, which I mentioned. All of them are related to abnormal organization of metabolites by the process of self-assembly. It could be cholesterol, it could be oxalate, it could be homocysteine, it could be uric acid, all kinds of molecules, metabolites that for some reason, which we try to understand, form these ordered assemblies. But understanding the fact that these structures could be formed is one thing. We would also like to inhibit this process. And we demonstrated also quite recently that we can inhibit the formation of the metabolite structures by polyphenols. This is EGCG, epicalocatechin calate, is a compound that is found in green tea. Tannic acid is a compound that is found in red wine. Both of them are known for support for some extent healthy aging, but we provide some mechanistic explanation. The fact that we see kind of polyphenols from plant source, which could help with avoiding the aggregation, the self-assembly of the metabolites, is relates to some hypothesis that a good friend of mine, Danny Otson from Denmark, suggested a possible connection between plant longevity and the absence of protein hybridization and basis for identification of aggregation inhibitors. So the reason that all these curcumin, green tea, ginger, red wine is good for us, it's not because the plants are producing these products for the health of the individuals who consume them. It's most likely that these structures were developed during evolution in order to provide some protection to the plants. And indeed, aging is normal, and even if you live long and you can do this pull-ups at the age of 90, sometimes by the age of 120, you will not be with us. All of us will not be here. But with plants, you can have plants which are thousands of years old. And we do think that it has to do with some of these compounds that these plants could make. So let me go back to the metabolites in the metabolome. As I said, these are end products of metabolism. There are so many in our body. And we have many thousands. We have more than 4,000 different metabolites. And this is the difference. I spoke about the identical twin. The main difference between people with exactly the same genetics is, but different health, different appearance, is mainly the composition of the metabolites, and metabolomics allows to, in a simultaneous way, quantify the thousands of different metabolites at a given time. And we call it metabolomics, but I guess all of us have done limited metabolomics. When we do a blood test and we test glucose, vitamins, hormones, cholesterol, we check not 4,000, but 20, 30 different metabolites. But metabolomic, the production of metabolites is so complicated. This is a map of human metabolic pathway. Don't worry, you don't need to understand it. It's just to illustrate the complexity. If you look at this very small part of this, if I enlarge it, zoom in on this, you see how complicated only this part of it. Each one of these names is a different metabolite that is made. So this is the entire map, and this is this small zoom in. So this is really a very, very complicated scenario. But imagine a situation in which you can do, in a low-cost blood test, check for 4,000 compounds, and do understand how it affects your health and how to create personalized profile for treatment of disease and understanding the correct nutrition, lifestyle in order to keep good health. So at the Blavatnik Center for Drug Discovery, which we established 5 years ago, we are very much involved in the development of new drugs. We already have one spin-off company, which is developing therapeutic treatment for certain type of cancers. We have other products, other projects that are on the verge of either being spun off into companies or licensed to a large pharma company. And all of you, when it will be possible to come to Israel, are invited to come to our center. It's also very nice. And also, even before that, you can do a virtual tour. You can come to our website, just write Blavatnik Center for Drug Discovery. And in our website, you will find a link to a virtual tour in the center. But understanding the importance of metabolites, the importance of metabolite medicine, we just established a new division to the center, a new division which will be involved in the study of metabolites and understanding it in a large complex. It will include profiling unit to recognize specific metabolic signature in samples from different disease states and others. A metabolite pharmacology unit to understand the mechanism of metabolite, and very importantly, metaboinformatics unit to understand the huge data that we will collect from different metabolites. And as always, we are interested with the collaboration with industry and having a crossover between the academia and industry. So this is from a paper to be published soon. Like the example that I gave, we can look at identical twins using our state-of-the-art machinery, understand the list of different metabolites, not only glucose and cholesterol as we do in normal blood tests, but thousands of different compounds, and do computational analysis, bioinformatics, analytic, crystallography, microscopy, in order to understand the function of such a collection of metabolites. And many times, if you look for, even in a simple blood test, when you have only a few tens of metabolites, sometimes you don't really understand the patterns, and and even qualified physicians do not understand everything. So it's a little bit like looking for a needle in a haystack. So this is why, again, from our paper, we are going to use big data and all kinds of computational machinery. And this is where metaboloinformatics get into the very important here to take together the data with databases, published data, and understanding experimental design, association between disease, structure and function, identification of biomarkers that are related to different diseases, and discovering new mechanisms of action. So this was a short presentation, but I would like to give you some take-home messages. So keeping biomolecules, not only protein as was thought in the past, but also other unassembled is a major challenge of biology, which is changed during aging. The aggregation, the self-assembly, the sociology of our molecules is correlated with major human diseases, and it's also correlated with aging and disease. We have now tools to know which metabolites tend to accumulate in different diseases, and we try to understand patterns of the metabolite aggregation and organization, and we try to control this process. And before we have very specific drugs, I would say that plant-based nutrition and other means may provide some way to reduce such molecular aggregation. This is my group over the years, not the current one. And many of them are already professors by themselves, and Tel Aviv University, Hebrew University, Bar Ilan University, the Technion, and also universities in China and India. I was speaking fast. I tried to show you a new direction. So everybody is welcome to contact me or to visit our website or both. And I look forward to meet you in person. And until that time, thank you very much. And I look forward to hearing from you. Thank you.
Hello, thank you. My name is TAL DVIR from Tel Aviv University. I'm the director of Tel Aviv University's Center for Nanoscience and Nanotechnology, and I'm also a researcher at the Department of Biotechnology in the Faculty of Life Sciences. And I will share with you now the Center for Nanotechnology at the university, and then a little bit about my work dealing with tissue engineering. So I will share my screen now. So the Center for Nanoscience and Nanotechnology at Tel Aviv University, it was established about 20 years ago. It was the first nano center in Israel. It has clean rooms and labs with a total area of 550 square meters, and it is supported by Mr. Fred Shaul, who helps us along the way with purchasing new equipment. We have a fleet of more than 60 systems, which are very advanced fabrication and characterization. We have a multidisciplinary expert staff, and we develop new processes of fabrication and characterization. And the center supports more than 100 academic research group, and it also supports companies and provides services to startup companies and even mature companies from Tel Aviv and all over Israel. We also have many collaborations, international collaborations, just to name a few, with Northwestern University, which is the #1 university in material science and engineering in the world, and Tsinghua University, the #1 university in China. So the new building, which is now under construction, you can see now at camera. And hopefully in 2 years, we will have the state-of-the-art laboratory building in Israel. And this is how it will look. It will have more than 8,000 square meters with 4 levels of advanced equipment and 600 meters of clean rooms with state-of-the-art facilities. And we have core members of the center in different fields. And just to name a few, from biophysics, we have Professor Roy Beck, drug delivery, Professor Dan Peer, who's also the Vice President for Research and Development, Photonics, Nanophotonics, Professor Kobi Scheuer, Regenerative Medicine, this is what I work on, Nanomaterials and Regenerative Medicine, Lee Adler Abramovich, Neuroengineering, Professor Yael Hanin, Molecular Electronics, Professor Shachar Richter, Nanosensors, Professor Fernando Patolsky, and Nanobiophotomics, Professor Yuval Ebenstein. And we're also recruiting new professors to the building, because this building will have the best researchers at Tel Aviv University working in one building in the field of nanotechnology. And I will now describe a little bit the research that we're doing at the center. For example, Professor Tal Ellenbogen from the School of Electrical Engineering is studying and controlling interactions of photons in nanostructured optical materials, and is doing it for different applications, from functional contact lenses to other things, energy and other things. Professor Yuval Ebenstein, his group is focusing on single molecule genomics. Professor Adi Aryeh is shaping electron beams and spherical aberration correction with nanoscale masks. Professor Dan Peer is working on drug delivery, state-of-the-art delivery of RNA molecules to treat different types of diseases. He's a pioneer in his field. And Professor Ehud Gazit from Life Sciences is also working on self-assembly of nanostructures, exploring biological and bio-inspired molecular self-assembly. And is not just working on the basic science, he's also working on different applications from biomedical to other fields. And with that, we will move on to the second part of my talk, which is the research that is done in my lab. And I called this talk Nanotechnologies for Mending Broken Hearts. And my lab at the Sagol Center for Regenerative Biotechnology, and also the Faculty of Life Sciences, Faculty of Engineering, and the Center for Nanoscience and Nanotechnology, is working on tissue engineering. We're engineering different types of tissues, from the heart, brain, spinal cord, intestine, kidney, and other tissues. But since I do not have a lot of time to discuss everything that we do in the lab, I will focus on the heart and the things that we do to regenerate the diseased heart. So before starting to tell you about the technologies that we develop in the lab, I will say a few words about the disease that we're trying to cure. So first of all, heart diseases are still the #1 cause of death in the Western world, more than all types of cancers together. And myocardial infarction or heart attack capture a significant fracture of these diseases. And it happens when a major blood vessel that nourishes the left ventricle is blocked. As a consequence, blood, oxygen, and nutrients cannot arrive to this area. The cardiomyocytes, cardiac muscle cells, are very sensitive cells. If they do not get enough oxygen, they simply die. And eventually there is a scar tissue in this area which cannot contract, cannot send pulses of blood to the rest of the body. And the poor statistic says that 50% of the people who've had a severe heart attack will die within 5 years. Currently, the only solution for this end-stage patient is heart transplantation. And since we all know that there is shortage in heart donors, there's a need to find new approaches to repopulate this area with tissue that can contract to regain the function of the heart. And this is exactly what we're doing in the lab. We're working on tissue engineering. So what is tissue engineering? What is the concept of this approach? We have a patient, as you can see here, with a defected organ on its left side. And what we do in the lab, we're engineering a substitute tissue in the lab that will have the same function, if we're talking about the heart, will contract nicely in the lab. And eventually we can remove the defected tissue, and under sterile conditions, as you can see here, we're transplanting the engineered tissue to regain function. And how does it really done? We can take cells from patients. We cultivate the cells to have enough cells eventually, and then we need to create tissues. And tissues in the body are not just the cells. The cells are very important. However, the biomaterial in between the cells is important as well. It is called the extracellular matrix. And what we do in the lab, we develop new types of biomaterials, allowing to accommodate the cells and allowing the cells to assemble into a functioning tissue, which can then be taken and transplanted back on the defected organ. And if we're talking about cardiac patches or cardiac tissue, engineering cardiac tissues, this is a typical cardiac tissue that we engineer in the lab. It is, the dimensions are rather big. This is a few centimeters in diameter, a few millimeters in thickness, and it contracts nicely in the lab for a long period of time without external stimulation. When we take these cardiac patches and transplant them, we can regenerate the infarcted heart in animals. As you can see here, we're elevating the ejection fraction. There's different technologies to regenerate the infarcted heart and to create patches. And one of these technologies or the approaches is to 3D print patient-specific tissues. And what we do in this approach, we look at the patient's CT, we look at the left ventricle, we isolate the blood vessels on the ventricles, and we transfer this scheme to the computer. We can add more blood vessels, and then we go to the 3D printer that we have in the lab, and we're printing patient-specific tissues from patient-specific cells and personalized materials. And this is how it looks. We can, this is a 3D printer, one of the 3D printers in the lab, and you can see that it prints cardiomyocytes, cardiac muscle cells with endothelial cells to form the blood vessels. And eventually what we're getting is a tissue that can fit the biochemical content of the person, the cellular content of the person, and also the anatomy of the patient, because it is made according to the CT image of the patient. And it is functional. You can see that it contracts nicely in the lab. You can see the transfer of the electrical signal transferring through the blood vessels that we create. And with that, we moved on to print volumetric tissues, such as whole hearts. As you can see here, we're using the patient own materials, the hydrogel that we process from the patient's own materials, and the patient's own cells to create a fully personalized human heart. It is a small-scale human heart, and this is how it looks in the middle of the process of printing. You can see the chambers, you can see the major blood vessels here in blue, and this is the end product, a human heart, a small-scale human heart that we next mature it in the lab with specialized bioreactors. We're circulating culture medium to nourish this heart through the blood vessels, and we're stimulating it to teach the cells how to speak with each other on the electrical level. And eventually we can take these hearts and actually transplant them in animals in a topic model, meaning in parallel to the natural art, not instead. This is the first step. The part that was printed in the lab, a human part, but in a rat. We can also print chambers or parts of the human heart, for example, atria. So we look at the atria, we create a model, we print it. This is how it looks, and it contracts nicely. I hope that you can see the contraction of this atrium. And we can then study conduction disorders in the lab. As you can see here, it contracts and it has disorders, and we now can use different types of drugs to evaluate or to assess what's the best drug to stop this conduction disorder. Another issue that we have with the engineered heart tissues is the transfer of the electrical signal. We're working with different biomaterials that perhaps do not allow the transfer of the electrical signal to be transferred nicely in the biomaterial. So if we use porous materials, we seed the cells can contract, these cells can contract, but they have a biomaterial in between that do not allow them to transfer the conduction. So what we do in the lab, we synthetically creating gold nanowires, incorporating them into the biomaterials to help the cells to interact with each other and to be synchronized. And you can see it here in these movies. This is without gold nanowires. You can see that the tissue is not contracting synchronously. But when we're using gold nanowires, everything is contracting synchronously. And this is extremely important when we want to regenerate the diseased heart. We can also create different approaches where we take gold nanorods and use them to convert energy, near IR energy, for example, allowing us to take these cardiac patches, put them on the heart, illuminate them with an energy, with a laser, and this will allow us to solder the tissue to the heart without the need for stitches. So this is what we do. We exploit gold nanorods and their physical properties to help us integrate these patches to the heart, and this is how it looks. You can see after 30 seconds of illumination, these patches are stuck to the heart and they're integrated very nicely. This is an electron microscope showing this is the heart and this is the patch, and it is integrated very nicely without the need for sutures, which can harm the tissue. We also create a cyborg tissue, and this is the integration of electronics with engineered tissues, not just because it's fun and cool to integrate electronics with living systems, because we want to evaluate what's the function of the tissue at all times, to evaluate whether this tissue contracts or if it contracts synchronously. So we created nanoelectronics, porous electronics, and we incorporated it with the biomaterials that we engineer in the lab to have a scaffolding material with many, many nanosensors within. Once we culture cardiac cells, we can immediately see on our computer what's the function of the tissue. If the cells contract, if they contract synchronously, we can add drugs and see how they react to these drugs. So it was a very nice publication, a Nature publication, a very nice patent for us, but it wasn't enough because in this case, we could sense the function of the tissue. However, if the tissue was not contracting properly, we couldn't do anything about it. So in the next step, what we decided to do was to engineer the second generation of cyborg tissues that, in addition to the recording, the sensing of the function, we can provide stimulation from afar. Just type on the computer and provide electrical stimulation, or type on the computer and release different types of drugs. So if you think about the technology, the patient is sitting in his house, is not feeling well. The physician can log on the computer, can sense the function of the tissue and decide how he wants to remotely activate the tissue. For example, provide electrical stimulation or release different types of drugs. For example, if it senses that there is inflammation, release anti-inflammatory drugs, or if it senses that there is not enough oxygen, release factors that promote the generation of new blood vessels. And these are things that I'm not imagining. These are things that we've done in this, we demonstrate in this paper. We can also create flexible and stretchable electronics for these tissues, and we can release different types of drugs in parallel. And when these drugs is terminated, we developed a new approach to reload drugs into biomaterials just by injecting the drugs into the circulation of to the bloodstream of these animals. We can also print 3D tissues with electronics within, and this electronics is now fully integrated. It is very soft and stretchable, as you will see in a second. It has the same mechanical properties as the tissue. So you can see now how soft this tissue is and how soft the electronics within this tissue. And you can see how robust it is. We can manipulate it. And eventually, we can sense the function of the tissue and provide electrical stimulation. And finally, I'm going to show the last slide. And what we do here, we're engineering now whole hearts, bionic hearts, with electronics all over the hearts that can activate the heart and release different types of drugs. And this is an ongoing project now in the lab that hopefully soon will be published in a very good journal. And with that, I would like to end up and acknowledge my group members who work very hard on the different projects. As I mentioned, we're working on hearts, intestine, spinal cord, brain, kidney, and other tissues, retina. And I would like to acknowledge my collaborators and my funding, and thank you very much for your attention.
Acabamos de escuchar 3 charlas súper interesantes en las cuales el foco estuvo puesto en la innovación. Por empezar, el doctor AVISHAY SINTOV, que es director del Laboratorio de Robótica de la Universidad de Tel Aviv, habló de los proyectos en los cuales está trabajando, que implica tanto el diseño así como el desarrollo de algoritmos que nutren a las máquinas. En este sentido, se focalizó en 2 iniciativas en particular. En principio, aquellas que están vinculadas con mejorar la interacción entre humanos y máquinas, de modo tal que esta interacción sea fluida y dinámica. ¿Para qué? Para que la inteligencia artificial pueda asistir a las personas en la realización de diferentes tareas del hogar, por ejemplo, u otras tareas diarias. También para que pueda ayudar a las personas que, ya sea por alguna necesidad o requerimiento especial, necesiten acompañamiento. También se habló sobre la impresión de manos robóticas y la utilidad que esto puede tener en diferentes ámbitos como despensas o fábricas, y también explicó que es muy importante tomar los datos que se obtienen a través de la manipulación que hacen estas manos robóticas para luego desarrollar sistemas de machine learning. A continuación, tuvimos el placer de escuchar al profesor Gazit, que es director de la Cátedra de Nanobiología de la Universidad y que habló sobre medicina metabólica. Por empezar, explicó que los metabolitos, que son producidos por el metabolismo y de los cuales hay miles en el cuerpo, en ocasiones, si están en una concentración demasiado alta o cuando forman ciertos autoensamblajes, pueden estar vinculados al desarrollo de ciertas patologías, como por ejemplo el Alzheimer, el Parkinson u otras enfermedades degenerativas. Habló en relación a lo que es la homocisteína y varios estudios que los han vinculado con infartos o enfermedades cardiovasculares en niños incluso. Y lo que destacó, que es muy importante, es que en la actualidad existen herramientas que permiten estudiar estos metabolitos en las personas con un simple análisis de sangre para tener un perfil bien claro y personalizado y así poder diseñar estrategias, conductas e incluso desarrollar drogas específicas y personalizadas para tratar o prevenir ciertas afecciones vinculadas con estos temas que estuvimos hablando. Por último, y también fue muy interesante, la charla del doctor TAL DVIR, que es director del Centro de Nanociencia y Nanotecnología de la Universidad de Tel Aviv y que también fue el primer investigador en lograr imprimir en 3D un corazón funcional. Esto fue un hito, esto es súper interesante y él contó cómo se está avanzando hoy en día en otras tecnologías o en optimizar incluso esta técnica para reparar lo que él dice corazones rotos. Así habló de la ciencia abocada a desarrollar soluciones para problemáticas vinculadas al corazón. En este sentido habló de la ingeniería de tejidos, que se basa en la toma de células de los pacientes que luego se cultivan en el laboratorio y se crean suerte de parches cardíacos, así los describió. Existen diferentes tecnologías para regenerar el corazón, una de ellas es la impresión 3D y es en la cual se está trabajando mucho en el laboratorio. Explicó que también esto permite tener, al tratarse de tejidos que se basan en células del paciente, tener un enfoque bien personalizado. Y explicó del desarrollo de nanocables que se incorporan a los biomateriales para que el tejido, en este caso estamos hablando del tejido del corazón, se contraiga sincrónicamente, lo cual es muy importante también para tratar o para lidiar con ciertas afecciones cardíacas. Destacó a su vez que esta electrónica incorporada al corazón no solo sirve para activarlo cuando es necesario, sino también para liberar diferentes tipos de drogas de forma personalizada según lo requiera el paciente.